Hello, welcome friends. Me Anila. The topic of my discussion is about digestive enzymes. But before this, don't forget to like and subscribe my channel and press bell icon. Friends, in this video, I will tell you how carbohydrates, proteins, fats, and other nucleic acids are digested by enzymes in human body. What are carbohydrates? These are compounds containing carbon, hydrogen, oxygen. For example, glucose is a carbohydrate, also called monosaccharide. Fructose is also monosaccharide. Disaccharides are sucrose and maltose, and lactose are disaccharides. Starch and glycogen are polysaccharides. So these are carbohydrates. Proteins are made up of amino acids. An amino acid consists of an amino group, a carboxyl group, a hydrogen group, and another functional group. Fats, normally triglycerides or lipids, that consist of glycerol and fatty acids. So these are carbohydrate, proteins and fats, and nucleic acids such as DNA, RNA, deoxyribonucleic acid or ribonucleic acid. All these are present in our food and are digested by enzymes in human body. In our human body, digestive system secrete enzymes that digest these biomolecules. Digestive enzymes are biological catalyst proteins that convert complex food molecules into simpler forms. So, biological catalyst means it speeds up the reaction and is recovered at the end of reaction. There are many enzymes. Some enzymes are for carbohydrates, some for proteins, some for lipids or fats, and some for nucleic acids. So, digestive enzymes convert protein into peptides or amino acids. Digestive enzyme convert carbohydrate into monosaccharide, that is glucose, fructose and galactose. And digestive enzymes for lipids convert lipids into glycerol and fatty acids. So, how works? The substrate is the substance, that is protein, carbohydrate or fats, on which enzyme acts. Enzyme and substrate complexes form. Product is released and enzyme is recovered at the end of reaction. So enzyme speed up the reaction. Our first enzyme is amylase. It is an enzyme for carbohydrates. Amylase. It breaks down carbohydrates, starch and glycogen into simpler sugars. It is secreted by salivary glands in the mouth and pancreas into the small intestine. It converts starch to maltose and then to glucose. So, starch and glycogen are polysaccharides made up of many monomers, many glucose molecules. So, amylase break down these complex carbohydrates into simpler sugars secreted by salivary gland and pancreas in the small intestine. Starch is converted to maltose and then to glucose. So the final product is glucose or fructose or galactose. So amylase speed up the reaction and it breaks down complex carbohydrates into simpler sugars. Maltase, another enzyme, as the name indicates, it will be involved in maltose digestion. It converts maltose into two glucose molecules in small intestine. It helps in the final step of carbohydrate digestion. So maltase substrate is maltose. Maltase react with maltose, form enzyme substrate complex. Product is released, that is two glucose molecules, and enzyme is recovered at the end of reaction. It helps in the final step of carbohydrate digestion because after this absorption of glucose molecules in the intestine occurs. Sucrase, as the name indicates, the substrate is sucrose. It converts sucrose, the table sugar, into glucose and fructose secreted in small intestine. So glucose is a monosaccharide containing aldehyde group. Fructose is a monosaccharide containing ketonic group. So sucrase, in case of sucrase, sucrose is substrate, glucose and fructose are products, and sucrase is enzyme. It is secreted in small intestine for the digestion of carbohydrates. Now lactase. As the name indicates, the substrate is lactose sugar, that is milk sugar. It converts lactose into glucose and galactose, that are monosaccharides. Lactose is disaccharide, is converted into monosaccharides secreted in small intestine. So these amylase, maltase, sucrase, lactase, these are enzymes for carbohydrate digestion. Now, I will tell you about the enzymes for protein digestion. Number one is pepsin. It converts proteins into smaller peptides secreted by stomach, and it is activated by HCl. Pepsinogen is inactive form in the acidic environment. Pepsinogen is converted to active form, that is pepsin. So in the stomach, pepsin is released in an inactive form, that is pepsinogen, but in the presence of HCl, pepsinogen is converted to pepsin in active form. This pepsin converts proteins into smaller peptides in the stomach. Now trypsin, this is also enzyme for protein digestion. It converts peptides into smaller peptides secreted by pancreas in an inactive form, that is trypsinogen, and converts to trypsin. So trypsin converts peptides into smaller peptides. So it continues the protein digestion. It is secreted by pancreas in an inactive form, that is trypsinogen, and converts to trypsin, an active form that converts peptides into smaller peptides. Peptide has to be converted to amino acids. This last step is occurred by peptidase. It is produced by intestinal glands and it converts smaller peptides to amino acids. So, intestinal glands secrete peptidase. Smaller peptides are converted to amino acids. Now, amino acids are ready for absorption. In case of carbohydrate, glucose, fructose, galactose are ready for absorption. And in case of protein, amino acids are ready for absorption. Now, the enzyme for fats is lipase. As the name indicates, lipase. It is secreted by pancreas. It converts fats into glycerol and fatty acids. So, it is secreted by pancreas. It converts fats into glycerol and fatty acids. So, these fatty acids and glycerol are now ready for absorption in the bloodstream. So, carbohydrate in the form of glucose, fructose, galactose, protein in the form of amino acids, fats in the form of glycerol and fatty acids, they are now ready for absorption in the bloodstream. Now, there are enzymes for nucleic acids. First of them is nuclease, secreted by pancreas. It converts nucleic acids, DNA, RNA into nucleotides. Pancreas secretes nuclease, nucleic acids, DNA, deoxyribonucleic acid, RNA, ribonucleic acid. DNA is composed of phosphate group, sugar, and nitrogenous base. RNA is composed of ribose sugar, phosphate group, and nitrogenous base. These are nucleic acids. Nuclease converts them into nucleotides. Nucleotides means a molecule consisting of nitrogenous base, sugar, and phosphate group. Nucleotidase, secreted by small intestine, it breaks down nucleotides into nitrogenous base, sugars, and phosphates. This enzyme is secreted by small intestine. It breaks down nucleotidase. It converts nucleotides into nitrogenous base, sugars, and phosphate that are ready for absorption. In this way, digestion occurs in human body. So in this video, I have told you that how carbohydrate, proteins, lipids, and nucleic acids are digested by digestive enzymes in our body, and how enzymes act on substrate to form enzyme substrate complex. Product is released and enzyme is recovered at the end of reaction. In this way, digestion occurs in human body. Friends, if you have any question, you can ask me on comments. I can help you. Thanks.